Thanks very much, Heather. I think first and foremost, just a big welcome to all of you. Thank you so much for coming to visit us. Hopefully what you heard this morning in terms of some of the advances we're making on the sort of earlier equation as far as the company's technology is concerned, really my job as part of this session is to really bring it home to the patient, right? So just a bit of background. I joined Recursion coming close to about 3 years ago. And, you know, from a background perspective, I've been in the industry for quite a while now, you know, so before I joined Recursion, I was at BridgeBio as a CEO of 2 of the subsidiaries, had good exposure to oncology and to rare diseases there. And then prior to that, a long stint in the Roche Genentech world across the globe, where, you know, we underwent a labor of love trying to create these franchises or medicines outside the field of cancer, primarily in neuroscience, ophthalmology, rare diseases. So it's a true pleasure when I stand here today and we talk about our amazing technology, our amazing maps, the insights that these are generating, which are amazing in their own right, but ultimately we have to run that ultimate experiment, which is, do these insights take us to the patient in a way where these medicines will make a clinically meaningful effect on the patient's journey? We talk about the concepts of industrialization. That's important in several respects. It's important to think about industrialization in terms of scale, and that's because the unmet need across a huge variety of diseases remains acutely strong. There are thousands of diseases today where patients do not have adequate solutions. So scale is important. Industrialization as it relates to speed. I'm sure as you walked into Recursion today, you saw some pictures. You saw some pictures of patients. And every year, we have to take some of those down for obvious reasons. So there is a sense of urgency with which we need to get our insights validated, translated, and converted into medicines to help these patients to move further. And then also another axis of industrialization as it relates to precision. You know, we talked about this morning, we went through, you know, presentations from Lena, from Imran, from Laura. As we're building, as we, you know, scaling up our ability to connect the dots in terms of the phenomics data, in terms of transcriptomics, industrializing transcriptomics, industrializing hopefully at some point proteomics, right? And get closer and closer to what will really move the needle in clinical care of a patient in a responsible way. And when I talk about that, is both the axis of efficacy, but also the axis of safety as well. So one hopes that the ultimate output from all the work we're doing here will give us more effective medicines that can really move that clinically important needle in patients. So before I launch into the clinical programs and give you, you know, some sense as to why we're excited about what we have in mid and late stage development, I just wanted to spend a little bit of time just giving you a preview on how we think about value creation through our pipeline at Recursion. The way, having spent a lot of time in the industry, I think of our pipeline as one pipeline. The pipeline that Recursion executes with our Recursion nodes, and then the pipeline we execute in partnership. Obviously, our mission is to decode biology to radically improve lives. And by definition, that means the ambition is huge across all the verticals of biology. But at the same time, we also have values that push us to getting medicines quicker to patients and to more patients. So we have to be pretty disciplined and pragmatic in terms of how we think about value creation internally and where we join forces with partners, especially in areas of intractable disease, intractable biology, such as neuroscience, fibrosis, where we have consummated some of the largest partnerships in industry, not even moving beyond the sort of sphere of tech-enabled drug discovery. But what we, you know, our collaborations with Roche Genentech, my alma mater, our collaborations with Bayer, this is a true partnership, not just for capital reasons, but bringing in the intellectual horsepower from both organizations to really try and move the needle as far as understanding the pathophysiology of these super complex diseases. So our focus in what we're very intentionally structured is to keep areas like oncology, precision oncology, rare diseases, wholly internal at Recursion. And as I represent the clinical development team today, we're very intentionally building capabilities and infrastructure to really welcome these great early stage programs that Laura just shared with you, Project Alpha, Project Gamma. We're building early stage oncology development capabilities, as well as a formidable translational medicine capability to really continue to hone in the axis of precision medicine and offering solutions to patients that move that clinical needle. So here's a snapshot of our pipeline. And the first thing I personally like when I see this pipeline is that I don't see a fourth-in-class anti-PD-1. I don't see a third-in-class JAK inhibitor. I don't see a 10th-in-class BACE inhibitor, right? And you can see the nature of our pipeline, which, if you actually take a step back and think about what we've heard this morning, this is the essence of our platform, to generate novel insights in biology. And if you fast forward that concept into how does this translate into medicine, it'll typically translate into first-in-disease or first-in-class approaches, right? So there's the nature of the clinical programs. It has made us, for example, partner very, very closely with the FDA, with the regulators, with the patient organizations to really make sure that we truly understand the patient disease journey here so that we can design ultimately, with the great clinical development team we have here, the right experiment in patients to tease out effectiveness. So we walked hand in hand here. 2 of the programs have been granted fast-track designation by the FDA, and this will enable us to have more seamless interaction and share data and make data-informed decisions as we move forward. At the same time, these programs represent a huge unmet medical need across the board where these patients, as we speak today, have absolutely zero medical solutions to help them. So let's launch into the programs. The first program, as you heard Dean mention as part of his work, and I'll connect the link there, and we can learn a little bit more about Chris's PhD thesis. But the first program, it gives me great pleasure to introduce our program, REC 994, in the treatment of cerebral cavernous malformations. Now, one thing I didn't mention as part of my introduction is that prior to joining industry, I spent time as a clinical neurosurgeon. I trained as a neurosurgeon. And when I think about diseases like cerebral cavernous malformation, NF2-driven meningiomas, I had a very different solution to offer patients for these diseases, right? It was a pretty crass solution in terms of either resecting the lesions or trying to ablate them using radiotherapy, which has got more precise as the field has moved on. But at the same time, I think the unmet medical need here, when you think about brain disorders, one of the basic things I learned as a neurosurgeon is that the brain is a pretty unforgiving organ in the human body. Anybody who's not welcome to the party. So you have parenchyma in the brain, you have cerebrospinal fluid in the brain, and then you have blood vessels in the brain, and that's it. Nobody else is invited to the party. So when you get something like a cavernous malformation, a brain tumor, an aneurysm, there's always trouble that gets created, right? Trouble in terms of vascular leakage, increased intracranial pressure, focal neurological deficits, inflammation that leads to seizures, headaches, behavioral changes. All these things come and become part of the patient's journey, which tends to be a miserable one. So here we have our first small molecule program that's a superoxide scavenger that's currently enrolling in a phase 2 trial. Here we've been granted orphan drug status both in the US and ex-US. And really sort of delving a bit more into the disease, the unmet need here remains huge. This is the first industry-sponsored program that's attempting to tackle these trouble-causing malformations. We've had investigator-led studies here. We have a study that's ongoing at University of Chicago that's looking at atorvastatin. And then we had a negative study that just read out in Europe, in Italy, using propranolol. So again, more disappointing news for the patients. Agents that perhaps showed some mechanistic promise haven't really moved that needle clinically that we care so much about. What happens with these lesions? These lesions are essentially mulberry-shaped malformations that have very thin walls, and really the problem is twofold. One, they have a finite risk of rupture that can lead to intracerebral bleeds, that can lead to high morbidity, quite often to mortality. The other integral part of this disease, which has been poorly understood by investigators, by clinicians, and also by regulators, is what happens when there's vascular leakage, when you start finding blood deposits around the actual lesions, and this starts a cascade of inflammation, and what sort of signs and symptoms does that lead to? So when I first came across this program and had to have regulatory interactions, the first interaction was with the CV Met division of the FDA, and they wanted to talk about stroke. Now, the stroke risk here is 0.4 to 0.6% on an annualized basis. And what we know about cerebral cavernous malformations is that it gives you a finite stroke risk, but that's certainly a small part of the patient's journey. And the CV Met division wanted to talk to us about stroke scales, MRS, and the National Institute Stroke Scale, which to us was not really fully appreciating what the patients were going through. So strategically, we eventually managed to pivot this program into the neurological division of the FDA, where, again, arm hand in hand with the Angioma Alliance, with the Patient Foundation, with some patients actually taking the regulators through the journey, we've had very robust discussions to date on what exploratory endpoints we could look at here. At the same time, both patient-reported outcomes as well as clinician-measured outcomes as well. So we're paving the ground here. Nobody's developed a drug. There's no regulatory precedent. There's no precedent in development for cerebral cavernous malformations. Is this a rare disease? I mean, you can look at the numbers there, right? I mean, the way we think about rare diseases, often we think about smaller populations. We've seen amazing progress and fantastic for patients to see advances in cystic fibrosis, more recently in spinal muscular atrophy area I was deeply involved in at Roche with the PTC Therapeutics deal there. But when we look at CCM, I mean, the numbers are impressive here, right? We have up to like 360,000 symptomatic patients today in the US and 5 key European markets. As MRI gets more frequently adopted into the evaluation of neurological disease, becomes more accessible, becomes cheaper, these numbers will only go up, right? Both in terms of incidental findings of CCMs, at the same time, the propensity of these lesions to eventually start causing clinical symptomatology. Today, given those caveats with regards to readily accessible imaging, it's approximately over 1 million patients today worldwide live with these lesions today. There's two forms of the disease. There's a familial form that's caused by aberrations of the genes CCM1, 2, or 3, and this represents roughly between 20 and 30% of the whole CCM pool. At the same time, as I mentioned, this is inherited in an autosomal dominant way, and when we compare it to the other diseases, the numbers here are exponentially large. Again, no approved therapy as it sits today. It's primarily supportive care. Patients have headaches. They have medicines for headaches. Patients have seizures. They have anticonvulsants to help control that. And the seizure frequency can vary. There's patients who have seizures on a weekly basis, on a daily basis. So it's a real crippling disease in terms of quality of life, ability to drive, and the things that we normally take for granted. So this is a good point for me to now delve into the science of this disease via introducing no one other than Chris Gibson to give you a little preview into his work that led to the formation of Recursion.
Thanks, Shaf. Thank you so much. I'll go through a quick summary of my dissertation, but before I do, I want to just talk about something really important here. Why is nobody else going after this disease? So the genes that encode this disease, or cause this disease, encode scaffolding proteins, which are not considered druggable by most folks. And in fact, I was at a conference a couple of weeks ago with one of the heads of research of one of the top 10 biopharma companies. And I was talking about this program, and he said, Chris, I love what you guys are doing at Recursion, but the reason none of us are going after this is because we know that no medicine is going to revert these lesions, these aneurysm-like lesions in the capillaries of the brain, back to the normal capillary. And I said to him, we don't need to do that. We don't think we need to do that. We think we need to slow the growth of these lesions. We need to reduce the number of these lesions over time, but most importantly, these are not high-pressure lesions. These are not lesions that are growing in the arteries or even the larger veins of the brain. These are growing in the capillaries. So the law of Laplace here does not apply so much as if these were real aneurysms growing in high-pressure vessels. In fact, what we need to do is actually reduce the activated sort of inflammatory milieu that surrounds these lesions. In fact, there's some fantastic papers that show in these patients, their symptoms arise most typically when they have some other comorbidity. So a patient might go for 6 months without seizures, and then all of a sudden they get the flu, and all of a sudden their seizures are hitting off because these lesions are inflamed, they get leaky, you get hemosiderin deposition, which leads to this feed-forward inflammatory milieu. And so we believe at Recursion, if we could start to reduce that activated state of the endothelium, one could actually go after this extraordinary opportunity to bring a first-in-disease treatment to one of the largest areas of unmet need remaining today in the rare disease space. So how did we get there, especially given these are undruggable targets? And unfortunately, I think probably like Rob called Dean, so he had to hop on a call real quick. I was going to make a lot of jokes about Dean and how bad he was in the wet lab. But what Dean was able to do in his work at the university was partner with a small number of other labs to really create a state-of-the-art of this field. And in 2011, this is what it looked like. This is from a Nature Neuroscience review. This is what people had figured out about CCM. And Dean's lab was one of the labs that helped uncover, one of the reasons he came to Utah, that CCM1, CCM2, and CCM3 are the genes that drive this disease. And in his laboratory, we had actually helped to hone this hypothesis using traditional molecular and cellular biology tools that the disease was being driven by activation of this protein called Rho-A. And so what we actually did was take a very, very simple approach to mitigate the activation of Rho-A using a really common drug called simvastatin, which through HMG-CoA reductase will mitigate the activation of Rho-A. And we put it into these incredible animal models that Dean's lab had built that are almost like mini CCM lesions just in a mouse, which is not always what happens in animal models. And we were so excited to read out this data. And I remember being in lab when Aubrey Chan, the student who was leading this program, read out this data. So after about a year of doing this experiment, we had statistically no effect on the lesions. There was a strong trend here, almost a statistically significant trend towards increasing the number of lesions in these mice. That was a devastating moment for us. We felt like we were on the cusp of making a real breakthrough for these patients with a medicine that was going to be extraordinarily inexpensive and hopefully have a benefit to them. But it caused us to reevaluate our process. And when we were doing that work, we noticed something. We noticed that when we modeled this disease in human cells, human endothelial cells, like the ones that are probably running through our pipeline right now at Recursion, the cells looked really different when you modeled the disease. You can see the difference yourself. You don't have to be a pathologist, but you can see that these cells look different. And so we wondered whether we could take an unbiased approach, whether we could get away from this traditional molecular and cellular biology approach and ask, is there some drug, regardless of its mechanism, that can make these 6 cells, SICCM2, look like the healthy ones, SI control here? And so we got to work. And because I was so bad in the laboratory, as Dean made sure to mention, I tried to use as many robots and software as I could to run this project. And so what we actually did was found some incredible open source software from a woman named Ann Carpenter at the Broad. She really pioneered this field. We got very small scale robotics, which was my hand still, but a 16-channel pipette, which was better than a 1-channel pipette at the time. And we started screening about 2,000 FDA-approved bioactive molecules or molecules that were commercially available that we thought could give us some insight into this disease. And let me show you what we found. So here's the same images, SI control, SICCM2. We want to make the green one look more like the less green one. For those of you who are purists in the field, you'll know Rho activation causes those green actin stress fibers. So, we knew that was happening. And look at simvastatin. Simvastatin was flagged by this cell profiler algorithm as a rescue of this disease. And you can actually see, if you look in the middle of those cells, there's less of those green tiger stripes. And this is actually, consistent with the hypothesis, but we'd already done the animal model experiment. We already knew that it wasn't working. Our algorithms also pulled out 2 other drugs. Cholecalciferol, I don't know if anybody's aware of this. This is vitamin D, which is like the only vitamin that's survived all the trials showing it actually has some utility in folks at higher than normal levels, and REC994. Now, what's most interesting to me is that, to my eye, neither cholecalciferol nor REC994 look to me like they're rescuing. And this was the scary part of my dissertation, because I had spent a lot of Dean's research money on this project, and the computer was giving us back hits that looked like they were not going to work. But we pursued, and we decided to finish the experiment and ask the question, was the computer seeing something that we had missed? And it turns out that it was. Because as we took both vitamin D and Rec-994 into these animal models, just a few months after the failed experiment with simvastatin, both of those molecules delivered a reduction in the number of lesions, both in mice driven by CCM1 mutations, but also in mice driven by CCM2 mutations. Together, those making up about 80% of the causative mutations of patients in this field. And what's more, when we did other preclinical work looking at this hypothesis of vascular stability, we found that there was a deviation in acetylcholine-induced vasodilation, which you can see here on the bottom left, which was completely rescued with Rec-994. We saw that there was a dermal vascular leak in the animals that you can see on the bottom right, consistent with what we know is happening in the brain with these vessels being leaky, which we could almost completely rescue with Rec-994. And it was these data that gave us the confidence not only to pursue this program, but actually for me to finish my dissertation, to leave medical school to start Recursion. And what's funny about Recursion is we didn't work on this particular program for 3 years after that. We didn't have the resources to go try and explore that first hypothesis I talked about this morning and to try and build a translational and preclinical team that could take the medicine to the clinic. But the founder of NPS Pharmaceuticals, who also is based here in Salt Lake, told me one day, he kind of put his arm around my shoulder and he said, you know, Chris, it's going to be important that you actually take medicines to the clinic at some point. You should probably go back to that compound and advance it. And so we licensed it from the university, and Recursion was really starting to get rolling on our platform. And so we were then in a position to advance this program forward. And so I won't go through all of the detail here, but suffice it to say, for those who understand endothelial biology, reactive oxygen species are a very well-proven activator of the endothelium. If you have a lot of reactive oxygen species in the endothelium, the cell-cell junctions break down, the cells become inflamed, and you get all kinds of things which are very consistent with what we see in the context of this disease. What this drug does is scavenges superoxide, which is the predominant reactive oxygen species that arises when you lose CCM1 and CCM2, which we were able to determine is driven by changes in both superoxide dismutase 2 and FOXO1 transcription factor levels. And we believe that if you can mitigate these effects, you can dramatically reduce the inflammation in these cells, leading to a more stabilized quiescent endothelium, which in the blood-brain barrier is so critical to help reduce that feed-forward inflammatory milieu. So I'm very excited to turn it back over to Shaf to talk about the phase 1 trial results.
Thanks, Chris. So just bringing it back to development. So you heard a little bit about the mechanism of action, what we think this particular drug is doing. And of course, our first step has to be to put it into a complex human system, in this case, healthy volunteers, and see how the drug behaves. As you'll hear as I go through the clinical programs, quite often when we put known or NCEs into the first healthy volunteer study, it's a moment of trepidation, right? Because these drugs have never been into a complex human system. So everything from how it's going to behave in terms of pharmacokinetics, in terms of exposure, in terms of tolerability, safety, all become areas of high vigilance for us. Here I'm very glad to report this drug has got a very benign profile. We dosed all the way up to 800 milligrams, both in the single and multiple ascending contexts. And you can see the numbers there, right? In terms of serious adverse events, absolutely nothing seen there, no drug discontinuations. So first hurdle, drug has got linear pharmacokinetics, great exposure. Certainly when we relate it to the levels we need to try and modulate the disease, we've got a large margin here to work with. Let's get to the clinical trial. So this is a super exciting clinical trial from my perspective. As I mentioned, this disease has never had anything in development, anything thought through with regulators in terms of what does the path forward look like here. Two things I want to point out about this trial. It's an exploratory phase 2 study that's really looking at a couple of things. It's looking at what has been measured before, things like the MRS, things like the National Institute of Health stroke scale. We know those entities, we know how they behave in disease states that are not quite CCM. At the same time, we've also done a lot of work in the background, really trying to understand the patient journey further. So here come in the patient-reported outcomes. Let's talk to the patients. Let's find out what they're going through. Let's find out what they're suffering from, okay? Headaches, seizures, focal deficits, double vision, behavioral symptoms, anxiety, stress on that, right? So let's try and get composite scales really validated. And the work we've done here has been with the University of Rochester to really come up with a pretty comprehensive 19-domain, 100-questionnaire patient-reported outcome, which we'll study as part of this disease, or part of this phase 2 trial. For neurological diseases, again, thinking about surrogate endpoints, here, this is not just standard MRI imaging. You know, Chris mentioned what happens pathophysiologically in this disease. You get this leakage, vascular leakage that leads to hemosiderin deposition. I'm glad to say that a consortium of investigators have done a lot of work validating a special protocol of MRI called the QSM, which is quantitative susceptible mapping, which is as powerful as an in vivo, a real-life in vivo study that really tracks from a temporal perspective, a time perspective, exactly what's the axis of this hemosiderin deposition and how does this affect any perilesional edema and inflammation. So we're measuring that in a subsection of patients as well. So we hope to get some sensitivity with the imaging modality there as well. We plan to enroll 60 patients, 2 dose groups, 400 milligrams placebo-controlled, and we're going to treat for 12 months, after which eligible patients will enter into a long-term extension study. Enrollment is progressing well, I'm glad to report, and we expect top-line data in the second half of next year for this phase 2 program. Moving on to the next clinical program, this is REC 2282 for the treatment of progressive NF2-driven meningiomas. This is an HDAC inhibitor that's a small molecule, has got some very special properties, and I'll talk through those as we compare this drug to the landscape of HDAC inhibitors. This is a unique trial. It's an adaptive phase 2/3 study. So it gives us a unique opportunity to be able to get, hopefully, an effective drug quickly to patients. Another highly underserved disease, brain tumors, malignant or benign. There are very few options out there. When we look at the brain tumors caused by mutations of the NF2 gene, meningiomas, vestibular schwannomas, are also known as acoustic neuromas, ependymomas, both occur in the brain and in the spinal cord. As we speak today, no therapeutic agents have been approved. There have been, unlike CCM, industry has tried to test different modalities, different anti-cancer agents to see whether we can move the needle clinically in these patients. So far, nothing has really met the mark. The disease itself is an autosomal dominant tumor syndrome resulting from an inactivation of the NF2 gene. And really, it's again like CCM, it's inherited either, it's autosomal dominant condition, and you have familial forms or sporadic forms as well of the disease. As I mentioned, there's different manifestations in terms of brain tumors, their locations, and this, the way I think about it is, you know, the actual disease itself, due to the loss of NF2 gene, gives us a real life cycle expansion opportunity. Our first forays into NF2-driven meningiomas, but mechanistically, and as far as the mechanism of action of the drug is concerned, we can certainly look at indication expansion into the other tumors I just mentioned, the schwannomas and the ependymomas. Standard of care is surgery. Now, again, coming back to my sort of neurosurgery days, this is probably the most tricky brain surgery to do for tumors. When one is conducting surgery for something like a glioblastoma grade 4, where the prognosis is dire, typically 6 months, one has more latitude in terms of risk-benefit, in terms of how you treat eloquent tissue, fully functioning brain tissue around the tumor. For meningiomas, these are benign tumors, but yet at the same time, to stop them from coming back, one has to excise up to a 1 centimeter margin of normal brain tissue. Now, I don't know how you guys feel, but if somebody was going to take 1 centimeter of my functioning brain tissue out, I'd be quite concerned, because usually you have collateral damage when that happens. So surgery comes with morbidity, often paralysis, often focal neurological deficits. And what we see in this genetic syndrome of NF2-driven meningiomas, often you get multiple meningiomas that show very heterogeneous behavior in terms of growth rates, right? So you can imagine the prospect of recurrent surgery is quite common here. So the unmet need is commensurately even higher. Presentation typically of an uninvited guest in the brain, as I mentioned before, a tumor in this case, that can lead to focal deficits, can lead to seizures, can lead to raised intracranial pressure that can lead to death in case of multiple large meningiomas, double vision, loss of visual acuity, certainly in the case of schwannomas where they're located, hearing loss is a prominent presenting sign there. Again, coming back to how many patients suffer from this, we estimate today to have around 66,000 patients who harbor these meningiomas, of which about 33,000 of them will be familial or will be sporadic meningiomas that have a demonstrated NF2 mutation. So the treatment population here is quite large as far as normal rare disease sort of numbers go. Primary intervention, as I mentioned, is surgical excision, and certainly a lot of the times these tumors occur in areas, at least from an anatomical standpoint, are not amenable to surgical excision. Getting skull-based meningiomas is quite common. Sometimes we see meningiomas that impinge onto the brainstem. That tends to happen, in which case focal radiotherapy is the only option to control these tumors. Let's get back to our operating system. How did we come across this? So in the early days, when we look at knocking out a gene, in this case NF2, and we look at our Prometheus plots that look at, okay, you can see in red here, these cells represent HUVEC cells where NF2 has been knocked down. And then we do mass phenotypic screening to see which agents actually revert these disease cells back to the green ones, the normal, healthy, functional state. And here we found REC-2282, a concentration of around 1 micromolar, very consistently brought these disease cells back to a healthy phenotype. So this gave us the first reason to believe that we have an agent here that could actually show promise in the treatment of NF2-mediated tumors. Mechanistically, loss of NF2 leads to loss of a key function called Merlin, a key protein called Merlin. Merlin is absolutely critical in tumor signaling. The pathways that are involved are the PI3 kinase, the AKT, and the mTOR pathways. And really what happens with aberrant mTOR, PI3K, AKT signaling is you start getting tumor growth. And HDAC inhibitors essentially block the protein-protein interaction with AKT and lead to suppression of this. So mechanistically it makes sense, the second reason to believe. We then came across and learned through an open-label phase 1 study that looked at both CNS tumors and peripheral solid tumors and looked at the effect of this particular HDAC inhibitor. I'll get to that data, but before that, we did additional profiling and we looked at the effect of this drug in traditional in vivo setting. So we looked at vestibular schwannoma xenografts in nude mice, and you can see that on top, you can see the, when we give vehicle, you don't really see an effect there, and then a very consistent effect in the growth of these xenografts and the suppression of growth of these xenografts when you give REC-2282. At the same time, if you look at a bioluminescence readout here in meningioma cells, you can see pretty marked suppression of the signal, and then of course upon stopping treatment, we see some durability of effect, durability of response there. And then we get to this phase 1 study I just mentioned. Here again, this study was not designed to provide statistical power of any sort. It was a signal-seeking study, but really encouraging to see two things here. One, we see that the NF2 driven central nervous system tumors, we saw a progression-free survival here of around 9 months compared to around 2 months for non-CNS tumors. And really what was encouraging to see for an HDAC inhibitor was that there were patients who were tolerating this agent for a duration of up to 27 months. So this gives us the second tag, the second hook to say, okay, we may have an agent here if we thread it carefully through a deserving patient population here, at least from a clinical disease standpoint, that we could have something that will give impact. A little bit more about this compound, REC-2282. When we compare it to, I think there's about 5 approved HDAC inhibitors in the market today, all have different characteristics. There are HDAC inhibitors that are given intravenously. There are HDAC inhibitors that are orally bioavailable. What we see in 2282 perfectly fits the ideal target product profile for what we would want for a CNS tumor. Orally bioavailable, targets the pathway effectively, orally administered, and has CNS exposure, and then hopefully no QT cardiac liability, which we consistently see with the other HDAC inhibitors. Let's get to the trial here. Again, this is the first in-class HDAC inhibitor for the treatment of NF2-driven meningiomas. What we require the patients to have here is MRI confirmation of the tumors. We're targeting both the sporadic form, in which case the patients need to have documented diagnosis of an NF2 mutation through tissue biopsy that could have been done through previous surgery, or a confirmed diagnosis of NF2 in familial cases. Outcome measures here are pretty standard for brain tumor studies. We're looking at progression-free survival at the 6-month mark. We're looking at tumor volumetrics as far as overall response rate, which will constitute of partial response and complete response, and we'll be measuring these tumors. What's interesting thing about this trial is it's targeting progressive meningiomas. So the patients will have to show evidence of a brain tumor that is showing progressive expansion. And the way the criteria have been set so far is they have to show 20% annualized tumor growth before they can get enrolled in the trial. Initial open label phase to enroll 20 patients, 2 doses, 40 and 60 milligrams, and then we'll undertake interim analysis and further FDA consultation on the confirmatory phase 3 design of the study. Enrollment is progressing well here as well, and we expect the interim analysis primarily targeting safety in 2024, next year. Getting away from the brain, now let's move on to FAP, our third clinical program. Here, this is an interesting story because, we've got now 2 indications that I'll talk about. Familial adenomatous polyposis, and then solid tumors, very recently harboring exon 1 and APC mutations. There's a lot of commonality as far as the pathway biology is concerned, which I'll touch upon. Here we have an inhibitor of MEK1/2, an oral small molecule inhibitor. And again, this program, with extensive consultation with the oncology division of the FDA, has garnered fast track and orphan drug designation status both in the US and ex-US. Another genetic disease or predisposition syndrome that leads to significant morbidity, often to mortality. Here we have an autosomal dominant tumor predisposition syndrome, caused by a mutation in the APC gene. It's a classical germline mutation that results in thousands, hundreds of thousands of polyps emerging in the GI tract. Typically, they start off when folks in the adolescence, the juvenile form that leads to hundreds of thousands of colonic polyps. So you can imagine, just not in addition to the risk of these polyps progressing to frank colorectal cancer, which is 100% if left untreated. At the same time, you can imagine that amount of disruption in terms of colonic function, what functional symptoms these patients have in terms of gastrointestinal transit syndrome, in terms of diarrhea, in terms of just general sequelae of inflammatory disease in the colon. As I mentioned, 100% progression to frank colorectal cancer if left untreated. We estimate today approximately 50,000 diagnosed patients in the US and in the 5 key European markets. No drugs approved to date. There was a fair amount of work done on celecoxib a while back, and certainly that drug was approved with the caveat that some key post-marketing studies were to be conducted, which never were, and hence the approval was withdrawn. So as we stand today, not a single agent or therapeutic class has been approved for the treatment of FAP. The option patients have is a rapid path to pretty mutilating surgery, which involves initially a pan-proctocolectomy to remove the susceptible lesions and to try and reduce the risk of progression to frank colorectal cancer. Also, these patients go through a pretty intensive schedule of surveillance, right, through endoscopy, sigmoidoscopy, pretty much on a 6-monthly basis to try and monitor the status of the disease. Again, coming back to some familiar pictures you've seen here, again, when we knock out in our system the APC gene here, and we get the disease cells due to the knockdown, and we screen. In this case, it was a pretty extensive battery of MEK inhibitors and ALK inhibitors that was screened here, up to about 1,500, I believe. And we certainly included agents such as selumetinib, binimetinib, and we found, even given that REC 4881 provided the most potent and specific rescue of these cells where the key gene APC was knocked down. So what's going on here from a pathway perspective? And this biology is common to both FAP and the Exin-1 program in solid tumors that I'll introduce next. And really here, the case is one of dysregulation here, right? We've got the beta-catenin in the Wnt pathway, the beta-catenin degradation complex. That's really a surveillance complex to try and stop activation of RAS. Activation of RAS lights up the MAP kinase pathway that eventually leads to the upregulation of MYC that leads to tumorigenesis. We see here that both Exin-1 and APC are key components of the beta-catenin degradation complex. And what happens with loss of function of Exin-1 or APC is that this complex stops doing its surveillance job and lets RAS continue its aberrant signaling and leaves the MAP kinase pathway unchecked to upregulation of MYC that leads to tumor formation. So any intervention that targets either elements of the complex through the beta-catenin or the Wnt pathway or targets the MAP kinase pathway has potential to actually arrest the growth of tumors here. In our case, we're targeting MEK here, a key component of the MAP kinase pathway, and this eventually leads to downregulation of MYC and suppression of tumor growth. Does this translate? Well, let's go to the in vivo studies, and this is probably one of the most impressive in vivo data sets we have here at Recursion. Here we have a validated, recognized mouse model. It's called the APC-Min mouse model, where the APC gene is knocked out. And you can see here, after 8 weeks of treatment, we see a potent reduction in polyp count. We've obviously benchmarked this against with celecoxib, one of the only other known agents that actually impacts this biology. And in a nice dose-responsive way, we actually reduce the polyp count across the board in this relevant genetic knockdown mouse model. What's even more impressive and excites me even more in terms of the potential clinical translation here is when we look at the graph on the right. Here, when we look at the progression of dysplasia, looking at the number of high-grade adenomas, we see with vehicle there's absolutely very little effect. Again, we see very little effect with celecoxib. But when we look at REC 4881, 3 doses, a nice dose response in the arrest of dysplasia. This is important because the reason these patients have colectomies and mutilating GI surgery is to ultimately reduce the chances of progressing to frank cancer. And so it's very reassuring for us to see we are reducing polyp count, but at the same time also reducing, downgrading the actual grade of dysplasia that occurs in these adenomas. What we did after that, which is the first time we're actually sharing this data, is run a dedicated phase 1 study to get a better sense of what the PK/PD is. I'm sure a lot of us here are familiar. Some of the challenges with trials in oncology is that often we don't have the recourse based on the mechanism of action of the target being modulated to go straight into healthy volunteer studies. In this case, it made sense because we needed to get a better sense for exactly what is the therapeutic index here, how is this MEK inhibitor tolerated, and what doses would give us the optimal reduction of pathway biomarkers such as phospho-ERK to then enable us to move rapidly into a phase 2 trial to make sure that we've got the dosing right here. So again, here we tested doses out, obviously checked for food effect, which I'm glad to report we did not observe a food effect here. But here, doses from 4 to 12 milligrams were tested in healthy volunteers. The drug was well tolerated. At the same time, we also ensured that we could match the data to what Takeda had generated before we licensed the molecule in. Getting to our trial, we initiated this trial in third quarter of last year. Again, from an enrollment criteria perspective, we're looking at patients who have a confirmed APC mutation, and we're targeting an aggressive disease patient pool here, patients who've already undergone a colectomy. So here the risk is that they will continue to develop polyps either in the rectal pouch or in the upper GI tract, in the duodenum, the ampulla of Vater, etc. And this is an aggressive form of disease because if you start getting polyps appearing post-colectomy in the rectal pouch, in the duodenum, the next line of surgery is pretty debilitating and happens pretty aggressively. So we have more pinpoint measures in terms of how this agent could potentially ameliorate the progression of disease. Part one of the trial will be looking at pharmacokinetic metrics, exposure metrics in these patients who've had their colons removed to absolutely make sure that the exposure, the PK, remains consistent with what we've learned in the healthy volunteer setting and to the data that Takeda generated, and then rapidly move on to the randomized phase with 136 patients across 3 doses, placebo control in part 2 of the study. We've made several protocol amendments here to do a few things here. This is a tricky population to recruit. We've had extensive consultation with our KOLs from the Mayo Clinic, from MD Anderson, with the FDA as to how do we make this study more fit for purpose for patients, most importantly, for clinical sites, for investigators to be able to recruit. So we made some recent protocol amendments here, and at the moment, we haven't recruited a single patient here as yet, but we hope to get sites initiated very rapidly. We have the first site initiated, and then we have a slew of academic sites where we think a lot of these patients who've had post-colectomies will present themselves for enrollment to the trial. So looking forward to the next update where we'll have patients who are actively enrolled in the study. We've also changed the protocol after extensive consultation. Initially, this was a 12-month study. We've reduced that to a 6-month period of treatment. So that should certainly ameliorate, some of the few delays we're having in terms of site initiation. And again, we'll provide an update, at a later stage on this program. Just bridging on to the same pathway biology, a program we announced fairly recently. And what's exciting about this program is it's still REC-4881, our MEK1/2 inhibitor. This time around, I get the pleasure of bringing in inference-based drug discovery, right? So as you heard this morning, how we go from maps to medicines, how we have these whole human genome level maps. We overlay small molecule perturbations on these, and very rapidly we can generate insights via inference and look at the potential utility of a drug for a particular mutation. Here again, the biology is common, dysregulation of beta-catenin, destruction complex. You saw in the previous diagram the critical role of APC and exin-1 in keeping that complex intact. And here, when we look at exin-1 mutated cancers, there's absolutely nothing, there's no solution for these patients out there. These often are highly resistant tumors, and when we look at across the whole spectrum of solid tumors, we go from lung to hepatocellular carcinoma, where exin-1 mutations as drivers are incredibly relevant. At the same time, then we look at endometrial cancer, bladder cancer. Overall, we're looking at a patient population of around 32,000 patients here. Again, happy to report that with all the advances made in genomic screening, we found that exin-1 is well represented today in all the industry panels. So in terms of patient diagnosis, we're working very closely with big data partners here to learn about both on the data and the sequencing side, which we'll need for this trial, as to where these patients are and how do we operationally bridge to run an effective phase 2 study. As I mentioned, this is a poster child from our pivot to inference. You can see here, I think the first reassuring thing and encouraging thing to see here is that both APC and exin-1 cluster together, right? So that's incredibly comforting to see with phenomics as the distal endpoint here. I think the second point to bring out here, which you see in blue, across doses from 0.1 micromolar to 2.5 micromolar, we see REC-4881 very potently rescuing the loss of function of APC or exin-1. So this gives us reason to believe that we have an intervention here with a well-profiled compound to enter into solid tumor study. As we speak today, we're actively in the design phase of this study, and we'll provide updates once we've homed in on a formal and final study design. Again, here we ran some validating in vivo studies here. You can see the waterfall plot where we look at mutant exin-1 mice compared to wild-type mice. And you can see a pretty reassuring tumor growth inhibition of around 70% in the mutant setting compared to around 50% in the wild-type setting. Certainly when you look at the related Kaplan-Meier survival curves, one can see the actual delta coming out quite nicely at a statistical significance between mutant and wild-type based on knockout of exin-1 or APC. So again, here from an update perspective, finalizing the design for 1B2 trial. We'll update when the trial design has been finalized. At the same time, we're also thinking about this agent in this context of solid tumors, both as a monotherapy, but also in combination with potentially different agents, potentially with checkpoint, with immunotherapy, in combination with chemotherapy, and in combination with other targeted agents. All these considerations are undergoing, rigorous evaluation as we speak. Finally, changing gears, getting away from tumors, moving on to what I'm proud to say, our first de novo Recursion operating system generated NCE, or novel or new chemical entity program, REC 3964, for the treatment of C. diff colitis. Here we have a small molecule, the first in class small molecule toxin inhibitor for Clostridium difficile that currently is in phase 1 healthy volunteer studies. Another serious disease, right? Clostridium difficile, I'm sure all of us are familiar with it. We've learned about it in terms of the socioeconomic impact it has, especially in elderly populations, in immunocompromised populations, in patients who are undergoing surgery, patients who are undergoing broad-spectrum antibiotic therapy. And really what happens is broad-spectrum antibiotic therapy essentially disrupts the microbiome and the gut flora that opportunistically enables Clostridium difficile to come in. What then happens is the toxins are released and the cascade of inflammation, localized colonic inflammation starts and has the potential to get systemic and make patients very unwell, both from a localized gut inflammation to toxic megacolon to systemic inflammation response syndrome that can lead to tractable levels of morbidity and mortality. Around 500,000, 500,000 cases seen for patients suffering from this disease. And really, the key, there's three aspects going here in C. diff development. There are agents that target the bacteria itself. We're all familiar with vancomycin, with metronidazole. More recently, there's been agents that target the homeostasis in the gut through modulation of the microbiota, through FMT, for example, that's still awaiting approval as we speak. Then we've also had large molecule toxin inhibitors that have also been approved and have had niche positioning, within the treatment paradigm for patients. What we feel we have here is a first-in-class small molecule toxin inhibitor, so far very easy to manufacture, very low cost of goods. At the same time, from what we're learning from the phase 1 study, has the potential to have very good tolerability and good druggability, as you'll see from the data that I share. Significant unmet medical need here with a large cost burden and socioeconomic impact. Recurrence of this infection occurs despite of antibiotic therapy. 20 to 30% of the patients recur, and a pretty tangible mortality. Close to 30,000 patients each year die in the US from uncontrolled Clostridium difficile colitis. Approximately 730,000 patients, US, 5 EU. And really here again, what gave us the reason to believe is, in this case, not a gene knockdown, but actually applying the C. diff toxin to both HUVEC cells and gut epithelial cells. And again, you see the familiar disease state cells when you apply this toxin, and then when you apply a series of molecules through phenotypic screening, we see nice reversion back to the healthy phenotype here in green. And you can see the difference between when this is very apparent to the naked eye when C. diff toxin is applied compared to healthy control. What's going on here mechanistically? It's a novel chemical entity, a new mechanism. So what happens here is the bacteria colonizes the gut. It releases toxin that gets endocytosed into the cytosol. What then happens is this autocatalytic cleavage that releases the toxins, glucosyl transferase enzymatic domain, into the cytoplasm of the cell. This then affects a key event in the pathogenesis of C. diff. What happens here is that it converts a set of row GTPases from the active state to the inactive state, and this then leads to key cytoskeletal changes, apoptosis, and dysfunction of the gut barrier that starts the inflammation cascade, and that goes unchecked unless the bacteria is treated, the toxin levels are diminished, or the microbiota is enabled again to try and remove the offending bacteria. What the drug does is it specifically affects the catalytic activation of the toxins glucosyl transferase, but not the host proteins. It's a very specific effect that we feel will have a real chance of actually stopping or impeding the cascade of inflammation that starts locally initially within the gut and then rapidly progresses on to lead to the pretty serious sequelae of colitis, megacolon, and then systematic inflammation as well. And you can see that here in terms of where the drug works in this key step of affecting the Rho GTPase is to convert them, to stop them being converted into inactive state. Again, the key question, does this translate, right? So we get to the models. Again, fortunate here to have a very relevant hamster model for C. diff infection. When we look at barrier integrity, again, in a nice dose-responsive manner, we see the agent restoring barrier integrity, which is key for overspillage of inflammation to not to occur. At the same time, then we look at some real-life functional data in terms of the Kaplan-Meier. You can clearly see the separation quite nicely of when the drug is given, and that extends the survival. That even extends beyond once the drug has been stopped as well. So this is reassuring evidence for us to then put the NCE into a phase 1 study in healthy volunteers, which is shown here. This trial is currently underway in the Netherlands. Both single and multiple ascending parts that have been initiated are well underway as we speak. And I'm pleased to inform a couple of things. One, what we're learning so far in terms of exposure, in terms of PK, in terms of safety, tolerability, we're seeing very encouraging signs so far, both of this NCE acting as a drug in a very dose-responsive manner, giving us linear exposure, PK, but at the same time showing very good tolerability up to doses of 1,200 milligrams, right? So those MAD portion is still continuing for that, and we expect both the safety and PK readout in the second half of this year. I think another interesting thing with the recent JP Morgan is we've had significant amount of incoming interest from some big pharmas on this program. So we'll initiate those discussions, and we'll see whether we prosecute the development of this particular agent ourselves or in partnership. Those will be good conversations to have. So with that, I will close the clinical section. Look forward to any questions during the Q&A session. But Heather, back over to you.